patterns of abundance, distribution, and diversity of life on Earth. And I would argue that even though we've been tackling these questions now for more than a century, we still lack a coherent and mechanistic understanding of what drives these patterns. And now that we're living in the context of rapid planetary scale change, ecologists are now increasingly challenged to not only understand current patterns of abundance and distribution, but also to project future ecological states as the environment changes, including what these changes mean for human well-being. And so in some sense, I would argue that the core mission of ecology is actually changing over the next century, moving from a science of understanding patterns to a science that's focused on making projections. And so I would argue that maybe one of the most important questions in this context that we need to answer is, how do living systems, including the human dimensions of these systems, change in the face of environmental change? And this is the sort of main guiding question that sort of guides my research program. But the problem, though, is that the foundations of ecology were built to understand patterns, not to project change, and certainly not to connect this understanding to human well-being. And so I was trained as a community ecologist, so I'll illustrate this problem to you with a classic model in community ecology, the Locke-Volterra competition model. Hopefully you remember this model from an intro ecology class. But if you don't, what you need to know is this is a foundational model. It's been around for almost 100 years and it's still widely used today. And this model is generally used to understand patterns in the dynamics of interacting species, but it was originally designed to apply only to a single environment. So what happens then if we want to use this model to understand changes in the outcome of competition between two interacting species as the environment changes? For example, if temperature changes. Well, we can imagine that if we're trying to make projections for how the interactions of two species might change as temperature changes, we might want to know something about how the dynamics of the individual populations of those two competing species are changing as the environment changes. And if we want to make projections of how those populations will change as the environment changes, we might actually need to know something about how individuals within each one of those populations is changing as the environment changes. And so what you can see then is that in order to make predictions in changing environments, we often need to understand processes operating at lower levels of biological organization. And I would argue that this is actually a general feature of developing a mechanistic understanding of change in ecology, is that it often requires connecting understanding of processes at another scale. And so in my work, I address this challenge of predicting change in biological systems as the environment changes, but across scales, all the way from cells up to populations, communities, and ultimately up to human well-being. And I do this as a quantitative ecologist. And in this, and I draw on a sort of a variety of tools and approaches to generate a coherent and mechanistic understanding of change. I harness and develop mathematical theory, which I use to develop hypotheses about the mechanisms by which biological systems respond to change. And then I test these hypotheses in two main ways. The first way is by using carefully designed experiments in the lab and in the field. And the second way I test predictions from theory is by synthesizing large-scale data sets from a variety of sources that cover large scales, both in terms of space and time, but also in terms of taxonomic diversity. I then harness data science tools such as simulation modeling to turn all these data into new knowledge that I use to test predictions from theory. And a common theme to all of my work is that I aim to contribute to a culture of open science. I make all of my data and code publicly available. I contribute to the development of open source software tools. And I also lead training initiatives and collaborations with various communities to share these tools with a wide range of people. And so today I'm going to address three main challenges that fall under this mission of projecting responses to global change. And I'm going to suggest to you that a solution to these challenges is to connect understanding across scales of biological organization. So the first challenge that I'll address is projecting the consequences of aquatic biodiversity change on human nutrition and health. The second challenge that I'll address is projecting the dynamics of populations in a warming world. And the third challenge that I'll address is how can we develop a general understanding of how living systems persist in fluctuating environments? Okay, so the first challenge is projecting consequences of aquatic biodiversity change on human nutrition and health. So one of the greatest challenges facing humanity is securing a sufficient and nutritious food supply. And for many of the world's 7 billion people, food security is an ecosystem service or a benefit that's provided by aquatic ecosystems. And reliance on seafood to meet nutritional demands is not distributed evenly across the globe. So I want to draw your attention to this, this box here in orange. This is the tropics, an area of high and increasing rates of nutrient deficiencies. These are also areas with high reliance on seafood for nutrition, and also high rates of marine biodiversity change. And so this raises the question then of how could biodiversity change affect human health and well-being? And we know, of course, from nutrition and health science that diversity in diets is a pillar of human health. But what we don't know is how human well-being is related to biodiversity per se. What I mean by that is that we don't have quantitative and mechanistic links that have been established between biodiversity change in natural systems and human health outcomes. And I would argue that's primarily just because the fields of biodiversity science and the fields of nutrition and health science have been sort of operating largely independently. And so what I did was to bridge the gap between these two fields to develop a mechanistic understanding of how biodiversity change affects human health. And when it comes to human health benefits from seafood, we first need to recognize that understanding these benefits isn't just a matter of understanding changes in yields. And the reason for that is that aquatic species are valuable in the human diet, not only as an important source of protein and calories, but also as an important source of micronutrients like calcium, iron, and zinc, and also essential fatty acids like EPA and DHA. And these micronutrients are critical. They're increasingly recognized as well when populations have insufficient access to these micronutrients. This is known as hidden hunger when populations have access to enough protein and calories, but not to these essential micronutrients. And this is increasingly recognized as a major contributor to the burden of disease in developing countries. And so we can measure the nutritional value of a particular species of fish in terms of the contribution of a particular portion of fish relative to recommended dietary allowance. And so if you've ever looked at the side of a cereal box, you're probably familiar with the concept of recommended dietary allowance, or RDA. This is essentially a metric that quantifies the amount of nutrient intake that's required to reach the requirements of a particular demographic of the human population. And so in this way, you can see that understanding the nutritional benefits that we get from seafood isn't just about optimizing or maximizing intake of any one of these nutrients alone. It's about reaching a range of nutritional targets simultaneously. So it's about optimizing this sort of multi-dimensional problem. So here then we addressed maybe one of the most important questions in ecology in the 21st century, which is, does biodiversity per se, that is species richness or ecological functional diversity, distinct from the identities or abundance of particular species, enhance human well-being? And so here we can draw on a really large and well-established body of theory in ecology known as biodiversity ecosystem functioning theory. And from this theory and hundreds of experiments, we have lots of evidence that increasing species richness is associated with increasing ecosystem function. And we can quantify the strength of this biodiversity effect here by fitting a power function to this relationship between species richness and ecosystem function. And the scaling exponent B here quantifies the strength of the biodiversity effect. And when we draw on this general theory and apply these methods, we can actually compare the strength of the biodiversity effect across a wide range of systems. So we took this approach here, but applied it in the context of human health and nutritional benefits that we get from seafood. And so here's what that looked like. So we could imagine two different scenarios here. You could imagine a scenario of high biodiversity in the diet compared to a scenario of low biodiversity in the diet. And biodiversity ecosystem functioning theory would lead us to predict that if species are complementary in their nutritional profiles, such that some species have high concentrations of one nutrient and low concentrations of another, such that there are trade-offs among nutrient concentrations across species, then biodiversity ecosystem functioning theory would lead us to predict that this complementary distribution of nutrients across species should lead to enhanced ecosystem functioning, or in this case, enhanced nutritional benefits. And so here we devised two new metrics of nutritional benefit. The first I'm showing you here on the left here, this is the number of nutrients or nutrient targets or RDA targets met per 100 gram portion. So biodiversity ecosystem functioning theory would lead us to predict that increasing species richness should lead to a higher number of distinct RDA targets met per 100 gram portion if species show these trade-offs in nutritional concentrations across species. Similarly, we predicted that increasing biodiversity in the diet should be associated with lower minimum portion size. That is that populations should be able to reach their nutritional goals more efficiently as species richness in the diet increases. And you can imagine that this is a benefit from the perspective of human health if human populations can meet the same nutritional targets but with less seafood biomass. If that is the case, then we would expect that increasing biodiversity in the diet should enhance human well-being because a greater proportion of the population will meet or exceed their recommended dietary allowances. So these were our predictions in hand from biodiversity ecosystem functioning theory. We tested these predictions by compiling a very large data set of nutrient and contaminant concentrations, as well as ecological traits for more than 800 seafood species. And in this data set, we focused mostly on wild species, but some species from aquaculture if they were mollusks. And for each of these species, we retained data only for which we knew which body part was included in the edible portion, as well as samples that were identified all the way to genus and species. And like anytime you pull together a data set from a large range of sources, you need to do some quality control. And here we subject all of these data to international standards for quality control, both in terms of the way that the data were originally collected and the way that they are processed. So with this large data set in hand, we created biodiversity experiments using a replacement design by simulating seafood diets at different levels of species richness. And this replacement design is important because it allows us to isolate the effect of species diversity from the identity of any one particular species. And we borrowed this approach from standard experimental design in biodiversity ecosystem functioning experiments like those done in grasslands and other systems. And the key feature here is that abundance declines as diversity goes up. So with these simulated diets following this replacement design, then we estimated the strength of the biodiversity effect by the ecological traits and their nutritional profiles. Okay, so what did we find? Well, let's first look at this, this yellow line here and this yellow dot here. So what I'm plotting here is the minimum portion size required to reach a given RDA target, and the yellow here is protein. And so what you can see here is that minimum portion size for protein is completely flat across species richness from 1 to 10 species, meaning there is no benefit of biodiversity for reaching protein nutritional requirements. In contrast, the micronutrients and the fatty acids have a different story. You can see that these other colors here, which correspond to the micronutrients and fatty acids, these are characterized by a negative slope, meaning that increasing biodiversity in the diet is associated with lower minimum portion size. And from the perspective of human nutrition, this is a benefit because it means much more efficiently. When we look at all 5 micronutrients and fatty acids simultaneously, like what we're seeing here in this purple line, we see the benefit of biodiversity is even stronger than for any of the single nutrients alone. And this is consistent with observations from other systems, and it's known as a multifunctional benefit of biodiversity. And the benefit of biodiversity is stronger in this case when we consider multiple functions simultaneously than for any of the underlying functions alone. We also found positive effects of biodiversity on the number of nutrient targets met per 100-gram portion. So as species richness increases here from left to right, a standard 100-gram portion that's composed of more species reaches more distinct nutrient targets simultaneously. And this is, of course, important from the perspective of human health. So one of the most exciting discoveries here is that we found that the distribution of protein among species in seafood assemblages is fundamentally different from the distribution of the micronutrient concentrations. So what I'm showing you on the left is the distribution of protein, and you can see it's fairly symmetrically distributed. In contrast, calcium is really, really highly skewed. You can see this distribution is really, really highly skewed, meaning that most species are fairly low in calcium, but there are some that are really, really high. And so what that means is that increasing biodiversity means that you're more likely to sample one of these really high performing species, and that leads to a benefit of biodiversity. And so what's really exciting here is that now that we've sort of discovered these two fundamentally different trait distributions, we can actually draw on a very large body of theory and evidence in ecology from what's called trait-based ecology, which is all about predicting how trait distributions will change in communities. And if we can predict how these trait distributions themselves will change as the environment changes, then this might allow us to predict how the benefits of biodiversity might change as the environment changes, for example, under scenarios of climate change. Okay, and so we also found that nutritional benefits are not only associated with species richness, but also another dimension of biodiversity known as ecological trait diversity. And so what I'm showing you here is the probability of reaching 5 micronutrient and fatty acid targets simultaneously per 100-gram portion actually increases as ecological functional trait diversity increases. So what that tells us then is that ecosystems that contain a broader range of ecological functional traits are also more likely to provide enhanced nutritional benefits. Okay, so to summarize, what we found here is that biodiversity is critical when it comes to meeting micronutrient and fatty acid targets, but not for protein. And biodiversity is essential to meeting nutritional requirements efficiently. And because we draw on this very general theory, we can then compare the effects of biodiversity to the benefits of biodiversity documented in grasslands and forests. And what we find here is actually that the benefits of biodiversity in this context actually are on par or exceed the benefits that have been very well documented in other systems. So the finding that biodiversity is essential to meeting nutritional targets efficiently is really important when we consider balancing sustainability goals for both human well-being as well as ecosystem health. And so what we've shown here is that protecting and restoring biodiversity will not only allow us to meet sustainable development goals, for example, the goal of zero hunger, but also protecting life below water. Okay, so now that I've shown you that biodiversity is critical, it enhances human well-being, the next question then arises, well, how can we project changes in biodiversity itself as the environment changes? And a key component to predicting biodiversity change under scenarios of environmental change is to project the dynamics of populations and population persistence under scenarios of environmental change. And so here, one of the key questions we need to answer is how will population abundance and distribution change as the environment changes? And one way we can tackle this problem is by studying metabolism. And the reason for that is that metabolism is a feature that's shared by all of life on Earth. Every living thing must uptake resources from its environment. It must then transform these resources within its cells and then allocate the products of this transformation to the various processes underlying maintenance, growth, and reproduction. And as ecologists, the rate at which all of these processes occur is what we call the metabolic rate. And one of the most widely observed patterns among ectotherms is that metabolic rates increase as temperature increases. And not only do we see this pattern across a very, very wide range of taxonomic diversity, but also the slopes of each one of these relationships consistently reflects the temperature dependence of cellular respiration, suggesting that constraints that are operating within cells might scale from cells all the way up to ecosystems. And this is what we call metabolic scaling, when constraints that are operating at fine scales emerge in higher order processes. And so there have been many efforts to scale up from effects of temperature on subcellular metabolism up to populations and communities. But all of these attempts to scale up have had to assume a relationship between temperature and population carrying capacity. And the reason for that is that population carrying capacity is a key parameter in many of our food web models. And so some of these attempts to scale up have assumed a positive relationship between temperature and population carrying capacity. Others have assumed a negative relationship, and some have assumed no relationship at all, but none of these have been tested empirically. And so we set out to test how population carrying capacity changes as temperature changes. And we can address this problem. We can make predictions for how population carrying capacity should change as environmental temperature changes by tracing population demography back to the energy budgets of individuals. And we can do this by recognizing that population growth is essentially an energetic process, right? In order for a population to grow, individuals within that population must acquire more resources than they need for maintenance, and then convert those excess resources into new individuals. So once we recognize that population growth is an energetic process, and we can understand how these energetic processes or metabolism respond to temperature, this allows us to then make predictions for how parameters, for example, of a population growth model might change with temperature. So one model that we can use to study population growth is the logistic model that I'm showing you here. And we use this model to predict how changes in population size as a function of parameters like population growth rate R and population carrying capacity here. So what we did here was to take this logistic growth model and recognize that underlying population growth is a set of energetic processes that we can measure via metabolism. And once we do that, we can then make predictions for how carrying capacity should change as temperature changes. And the prediction here that we're testing that comes from metabolic scaling theory, I'm showing you an equation form up here, but in words down here. And what this prediction tells us is that as mass or body size and temperature increases, individual metabolic demand increases and carrying capacity, K, decreases. And this might seem kind of counterintuitive. Why should carrying capacity decrease as temperature goes up? Well, if you can imagine a scenario in which resource supply is held constant and each individual has higher metabolic demand, then a collection of individuals with higher metabolic demand and the same resource supply will be able to support fewer individuals, right? So at equilibrium, population abundance is predicted to decline as temperature increases due to these higher individual metabolic demands as temperature goes up. So I'll just show you this prediction here graphically. What this prediction says is that as organismal metabolic demand goes up, as temperature increases, this leads us to predict a decline in population abundance as temperature goes up. And this decline in population abundance should be quantitatively predicted based on the increase in metabolic demand as temperature increases. So we set out to test this prediction empirically using an experimental system with phytoplankton. This is a species of green algae that we isolated not far from here off the west coast of Vancouver Island. And this experiment was done in collaboration with a whole bunch of awesome undergrads that I'm showing you here. The design of the experiment was fairly simple. We grew populations across a temperature gradient across 6 different temperatures. And I'm showing you this video of Tetrasalmus tetrahedrale here because you can see it sort of zooming around swimming. We chose this species because it's highly motile, like you can see here, and that allows us to meet the assumptions of a logistic growth model, which is that all individuals have equal access to resources. So with this experimental system in hand, then we first, though, need to make to measure how metabolic demand increases with temperature. So the prediction from metabolic theory is that metabolic demand should increase as temperature increases. We actually measured metabolic rates of these individual phytoplankton cells and found that, consistent with wide range of observations, that organismal metabolic rate increases as temperature goes up. That's not surprising. That's what we expected to see. But with this slope in hand, then we can then draw on this theory to make a prediction for how population carrying capacity should decline as temperature goes up. So here's our theoretical prediction shown here in the dots, and here's what we found. So what our experimental observations are shown here in the circles, and so you can see that population carrying capacity does decline as temperature goes up from left to right. But this decline was not quantitatively predicted by theory. Our theoretical prediction is in the dotted line. So what this means is that populations at these warm temperatures were able to support more individuals, higher abundances than theory predict. So our attempt then to scale up from our understanding of how organismal metabolic demand constrains a population level outcome didn't work. We were not able to quantitatively predict this decline. So either the model is wrong or there's something going on that we didn't account for. And what we observed over the course of this experiment is that body size, or in this case cell size, decreased with warming. This shouldn't have been surprising. It's also consistent with observations across ectotherms, and it's known as a pattern that we call the temperature size rule. So we observe this decrease in body size with warming. This is a phenotypically plastic response. But our original theoretical predictions, like what I showed you earlier, they don't account for this phenotypic plasticity, this change in body size itself with warming. And so these original metabolic scaling theories don't allow for the temperature size rule. You can see that mass here doesn't depend on temperature directly in any way. So what we did was to expand this theory a little bit to allow mass itself to depend on temperature so that we can account for this temperature size rule response in our predictions. And that's shown here. And so when we do that, and when we incorporate the temperature size rule or that phenotypically plastic response in our predictions, then we can actually quantitatively predict the decline in abundance over this temperature gradient. And so what I'm showing you here in this dashed line is the theoretical prediction that accounts for the temperature size rule. And what you can see is that we can quantitatively predict the decline in abundance based on the temperature dependence of photosynthesis or metabolism in this population of green algae. And so this is really exciting because it's the first evidence that change in population abundance with warming, those changes are mediated by increased individual metabolic rates. And the reason why that's exciting is that we can now harness our understanding of how individuals are responding to changes in temperature, how individual metabolic rates are responding to changes in temperature, and harness that understanding to make projections at the population level in terms of how populations will respond to changes in temperature. So I've now shown you that those metabolic rates at the individual level can predict population responses to changing temperature. But I would argue that any form of projection in changing environments needs to consider evolution. And the reason for that is that we now have quite a lot of evidence that rapid adaptation can actually occur on ecological timescales. So if we're trying to make predictions then of how populations will change into the future as the climate changes, it's critical that we account for contemporary evolutionary change in those metabolic traits. And so here we asked, how do those metabolic traits evolve in changing environments? And most of what I've talked to you about so far is changing environments in the context of changing temperature. But Of course, we know that concurrent with changes in temperature will be changes in resource supply. And so here we used experimental evolution to answer this question of how metabolic traits evolve or adapt in response to rapidly changing resource conditions. And so the resource conditions that we addressed were contexts of nitrogen limitation, phosphorus limitation, and light limitation. And after this period of experimental evolution, we quantified evolutionary change in some key metabolic traits. And the metabolic traits that we looked at are the minimum resource requirements. These are also known as R star for those of you who study competition. R star are these key traits that link physiological performance to the outcome of competition between species. And so going into this experiment, we expected, based on evolutionary theory, that we would observe trade-offs. So what I mean by that is that there would be adaptive trade-offs, in the sense that adapting to one environment would come at a cost of adaptation in another environment. And so in this case, a lower value is better, because the trait that we're looking at is minimum resource requirement. So a lower minimum resource requirement is better from the perspective of population. And so we would expect that a trade-off would look something like this, where getting better in one trait dimension, like on this horizontal axis, would be associated with getting worse on another trait dimension, like what's shown on this vertical axis. And what we would see is that populations after this period of experimental evolution would fall along this diagonal here that would represent an evolutionary trade-off between adaptation in multiple environments. So we predicted trade-offs in performance in these multiple in these changing resource environments, and what we found is the complete opposite of that. We found no evidence for adaptive trade-offs whatsoever. Remember, we were expecting to see a negative slope. We actually see a positive slope. And what we're seeing is that multiple metabolic traits are improving simultaneously in these rapidly changing environments. And so you can see that a large number of the populations, after exposure to experimental evolution in these different resource environments, improved in multiple trait dimensions simultaneously. And so we think that this is because these metabolic traits are actually intrinsically linked via a metabolic network, right? So you could imagine that if a population is subjected to low light, then it might invest in more efficient chloroplasts to harvest light. And we know that chloroplasts are nitrogen rich. And so you could imagine then that exposure to low light could also come with added benefits of using nitrogen more efficiently, and that would reduce your nitrogen requirement. So what we're finding here, though, more broadly, is that we're seeing these sort of simultaneous improvements in core metabolic performance in rapidly changing environments. And I think that's because in many cases, when populations are subjected to these very, very quickly changing environments, they're likely very, very far off some sort of fitness optimum or some sort of optimality frontier. What I'm plotting here is the Pareto frontier. This is to borrow a concept from engineering and economics. And it's only at this frontier, this optimality frontier, where we might expect to see trade-offs, that is, improving in one trait dimension would come at a cost of losing performance in another trait dimension. And so I think the key finding here is that our evolutionary theory, which leads us to predict trade-offs and sort of based on these assumptions of fundamental constraints, might not actually hold in these very, very rapidly changing environments when populations are very, very far off fitness optima. And I think this is actually suggesting to us that we might need to fundamentally rethink some of the assumptions that go into our predictions of evolutionary change in rapidly changing environments. Okay, so I now want to address the final challenge for today, which is how can we project species distributions in variable environments? And one really common way to make projections for how species distributions might change as the environment changes is to scale up from estimates of the fundamental thermal niche or thermal performance curves to patterns of species distribution. But one really important finding that comes from Jen Sunday's work from about almost 10 years ago now is that she found that species distributions often cannot be predicted from the fundamental thermal niche measured under constant lab conditions. So what she did was to compare the fundamental thermal niche with observed species ranges and found that species are often either underfilling or overfilling their fundamental thermal niche. And that sort of prompted a whole bunch of questions about what could explain these discrepancies between the fundamental thermal niche measured in the lab and observations of species ranges in nature. So could it be things like species interactions or other sort of niche axes that weren't considered, like water availability, for example? But one really important thing to consider when we attempt to make predictions for population persistence and range shifts when we're dealing with these nonlinear thermal performance curves is how the nonlinear nature of these curves interacts with natural patterns of environmental fluctuations over time. And this is due to a tenet of nonlinear averaging known as Jensen's inequality. And what Jensen's inequality tells us is that when we're trying to predict performance in a temporally thermally fluctuating environments, because this curve is nonlinear, we actually expect deviations from performance at mean temperatures. Specifically, when this curve is accelerating in this blue portion here, we expect that variation should increase performance relative to constant lab conditions. And in this portion here, variation should decrease performance relative to constant lab conditions. And the key thing here is that people are often using these thermal performance curves measured under constant lab conditions to predict species distributions under naturally varying conditions. So if we don't explicitly account for the nonlinear nature of these curves, we're probably going to get our predictions wrong. And so this led us to ask this question of how does thermal variability essentially alter what we might expect the realized thermal niche to look like? So we did a very simple lab experiment in which we grew phytoplankton under these thermally fluctuating conditions and measured their population growth rates and compared those to constant conditions. And what we found is that, not surprisingly, temperature variability systematically and predictively alters the realized thermal niche. So this green curve here is the thermal niche measured under constant conditions in the lab. This is sort of your standard thermal performance curve. When we introduce some amount of thermal variability in our lab experiments, we predict a shift in the shape of the thermal performance curve just based on nonlinear averaging, as I'm showing you in these dashed lines. These are our predictions. And then the orange line is our observations. And we see actually that we can quantitatively predict a shift in the thermal performance curve just based on nonlinear averaging over this thermal performance curve in constant conditions. And the key thing here is that what you can see is the entire thermal performance curve is shifted down and to the left. And when we look at this sort of pattern across a broad range of phytoplankton species, so what we see is that T-opt, or the optimal temperature for performance, most often shifts to lower temperatures than would be predicted based on constant lab conditions. So we made these predictions here by using thermal performance curves from about 100 different species of phytoplankton and their natural patterns of environmental variability where they were collected. And so predominantly we see these shifts in the thermal performance curve or the realized thermal niche to lower temperatures, except for a small number of positively skewed thermal performance curves in our data set, which show the opposite pattern. So the key thing here is that accounting for natural patterns of environmental variability actually causes a systematic shift in our expected realized thermal niche. And when we account for these natural patterns of thermal variability, we actually see that could potentially explain discrepancies between the fundamental thermal niche that we measure in the lab and species distributions. And so And so this could potentially explain those patterns of overfilling and underfilling that were originally observed in the absence of any sort of other species of those deviations, and really sort of highlights the importance of accounting for natural patterns of fluctuations anytime we're attempting to scale up from organismal performance to estimates of population performance in naturally variable conditions. Okay, so then in the last section of this talk, I'm going to address a slightly different way of thinking about living systems and population persistence in fluctuating environments. And I would argue that most of the way we think about biological responses to environmental fluctuations in ecology and physiology are based on some sort of feedback. And so we could imagine here that if we have some sort of environmental fluctuation, like I'm showing you in the gray line, it could be a fluctuation in temperature, like I just showed you. We often study population dynamic responses to environmental fluctuations based on a model of feedback, where the system is responding to the change, but there's some delay in that response. In contrast to that, there's actually a whole other class of biological mechanisms that enable living systems to persist in fluctuating environments that rely on feed forwards. And feed forwards have been studied in other sort of contexts, drawing on concepts from systems biology. But the key benefit of a feed forward, which I'm showing here in this right panel, is that they allow the system to capitalize on some environmental cue that happens in advance of the environmental change. And that allows them to prepare themselves in advance for that impending future environmental change, and that confers some sort of fitness benefit. So I'll explain that to you a little bit more here, just by pointing out that feedbacks are probably very familiar to us. They are reactive. So an example would be thermoregulation in endotherms. These rely on some sort of internal set point. When the system deviates from that set point with some delay, there's some sort of mechanism that comes in to counteract that deviation. The system then returns to the set point. In contrast to that, feed forwards are proactive, and they rely on some external environmental cue that the system capitalizes on to predict the future. So an example of that is this copepod here that's doing a diel vertical migration. It capitalizes on some cue associated with change in light availability at the surface. It then moves to depth to avoid higher predation risk at the surface when the sun is up and those sunlit waters are lit and visual predators can more efficiently find their prey. So the key thing here is that these copepods are capitalizing on some environmental cue, in this case, in terms of light, a change in light availability at the surface, getting out of the way. This allows them to prepare themselves and avoid predation at the surface. And then this confers some sort of fitness benefit because it reduces predation risk. So feed forwards arise more generally as organisms exploit repeated associations between correlated environmental variables with some sort of time lag to anticipate change or predict change. So I'll give you an example of that here. If we imagine that you're an E. coli and you're living on some sort of surface in the environment, and you're exposed to some set of environmental changes shown here in this these blue bars, you can see that actually this sequence of environmental changes, like a change in oxygen, for example, is actually highly unpredictable from the perspective of the E. coli. There's a really wide range in the distributions of these time lags, these taus. The same for this gray variable here. It also shows a really, really wide range of distributions in those lags between successive events. So from the perspective of the E. coli in an environment that's characterized by these changes in oxygen, those changes in oxygen are fairly unpredictable. But what you can see is that this blue event, which is a change in oxygen, happens always very closely after this gray event, which might be a change in temperature. So what this tells us then is that if the E. coli can capitalize on this correlation between a change in temperature that happens very quickly before a change in oxygen. It doesn't need to predict a change in oxygen. It can actually just detect a change in temperature. It can, for example, switch its metabolism from an aerobic to an anaerobic pathway in advance of a drop in oxygen. That allows us to prepare in advance, which confers some sort of fitness benefit. And then, of course, the problem is when this correlation breaks down and the cue becomes unreliable, then we run into all kinds of interesting problems. And so feed forward, these sort of cue-based feed forward processes are everywhere in ecology. They show up in phenology, in circadian rhythms, in those diel vertical migrations that I just told you about. And something that I'm exploring now is what I think is a major unexplored dimension of global environmental change, which is that these cues that organisms rely on to anticipate future environmental conditions might actually be changing as the environment changes. And I think that we need to get a good handle on how these cues are changing if we're going to make accurate predictions of how populations and communities will respond to changing environments. Okay, so to summarize what I've told you about today, I sort of started off with this major scientific challenge, which is how can we project future ecological states in this context of rapid planetary scale change? And I suggested to you that a solution to this challenge would be to study mechanisms that cross scales, for example, from the subcellular to the individual to the population scale. This requires us to connect understanding across boundaries and across scales, and also to use established tools in new ways. And I would argue that when we use this approach, this is going to allow us to fundamentally advance our understanding of living systems and ultimately to improve our ability to secure a sustainable future for humanity in the context of these rapid planetary scale changes. And with that, I would like to thank my funding sources and collaborators, and I would be more than happy to take any questions. And if you would like to reach me, you can contact me here.
Thanks so much, Joey. That's a fantastic connection of a whole suite of different topics. Do we have any questions for Joey?
I just have a, I do have a question. Thank you, Joey. That was awesome. That was amazing. I love the solid kind of theoretical kind of foundation, and then where you've gone with it. Congratulations. I was wondering about this, the biodiversity, the first bit of your talk, how, because obviously there's a latitudinal biodiversity gradient, we would expect that human health would be better near the tropics. Is that something that is observed? Either that, or we eat more in higher latitudes, I guess. Yeah, have you looked into that or thought about it?
Yeah, that's a good question. I mean, if only it were that simple, it would be a lot easier to predict these changes. I mean, not to say that observation is a really good one, of course, that we have these latitudinal biodiversity gradients. The problem is that human health and human nutrition, there's like, substantial disconnects between the biodiversity out there in nature and the actual health status of human populations. And unfortunately, as it turns out, many of the populations in those sort of tropical coastal regions are actually the most, have the highest rates of micronutrient deficiencies, for example. And they're also experiencing really rapid biodiversity change. So in spite of the fact that we might think that those ecosystems do provide benefits, and the benefits are, we can clearly document, the challenges that those areas are also experiencing, these high micronutrient deficiencies, could be due to access to the biodiversity that exists there. So I think it's really interesting what you're raising, and I think there's this really there's really a whole open field of questions that has to do with biodiversity change, and then access to biodiversity, and then how that actually shows up in the diet. For example, we know that human populations that rely on coastal species can often consume fairly high amounts or consistent rates, even as abundance is declining, and it requires a very, very large decline in abundance before you see that show up in the diet. So it's not always non-linear. I guess where I'm going is it's not always linear. It's often non-linear, the relationship between the change in biodiversity and the actual effects on the diet that you might see.
Right. Yeah. No, that's why I was asking, because I did suspect that that we do see those micronutrient deficiencies in the tropics. So why is that, right?
Yeah.
But yeah, no, it's very, very interesting. Congratulations.
Yeah. Thank you. Yeah.
Follow up on that, were the species pools that you constrained, were they by kind of biogeographic region, or do they just mix from the whole data set?
Yeah, that's a great question. We did it both ways, and I didn't get into showing you that, but I could pull up those figures if you're interested. So the challenge is that having high quality diet data for populations around the world is very, very tricky. And so, there's tons and tons of research that goes into these dietary recall surveys, basically documenting what people are actually consuming. So in the absence of that at a global scale, we for sure do not have that at a global scale. We do have that in some regions. We looked at traditional indigenous diets in North America, and there we're able to get groups of species that are traditionally consumed as part of traditional indigenous diets. Those are fairly locally constrained. And actually when we do the same analyses, but at the local scale, when the species pool is locally constrained, we do see similar effects of biodiversity, but they're actually weaker than what you see when you draw from the whole global species pool. And that's not surprising. That's associated with lower, higher what we call nutritional functional redundancy, meaning that species that are caught locally are more redundant nutrition wise than when we draw on the whole global pool. So there are differences, but those biodiversity effects do persist even at the local scale. They are weaker though than at the global scale. Yeah.
Alex?
Yeah. Oh, I can't hear you, Alex. I'm sorry. I don't know if others can hear Alex, but no. I can't. Okay.
Can you type it in?
If you want, you can type it in the chat. If, I can look at the chat and try to answer that way.
I have a question while Alex is typing that in. Yeah, I was just wondering if when you look at the local scale species pool, do you see that complementarity in the micronutrients, like the trade-offs, or is that only at the global scale?
Yeah, we do see those same patterns and complementarity. At the local scale. I can't give you like a quantitative estimate of how different they are. I think the patterns are similar at the local scale, but how much different they are. Yeah.
Thanks.
But yeah, it's a good question.
Oh, I'm muted. Alex, are you gonna give the typing a go?
No.
No.
Okay.
Away from keyboard. Okay. Do we have any other questions? Okay. I'm not picking up anymore. So with that, we can thank Joey one more time for dedicating part of her Tuesday evening back in BC to coming to speak to us about this broad array of research. And yeah, thanks so much, Joey. If you're ever coming through Sydney when the pandemic lifts,
yeah,
we're around. Hopefully you get to visit Yale sometime soon.
Yeah, I hope so too. Yeah, and I would, yeah, I look forward to the chance of meeting you all at some point in person. Hopefully we cross paths at some point. And yeah, if anybody has any other questions about any of this, just feel free to shoot me an email. I'm more than happy to follow up that way as well.
Awesome. Thanks so much.
Yeah, thanks everybody. And thanks, Erin, for the invitation.
Not a problem.
Okay.
Bye.
Bye, everybody.